So a chromosomal translocation is a rearrangement of parts between two non-homologous chromosomes. And recall that chromosomes occur in pairs, and those pairs are called homologous pairs. So non-homologous chromosomes are not members of the same pair. Now, several types of cancer are caused, at least in part, by translocations like this. And these are acquired in the somatic cells and are not present in germline cells. And so while translocations were first recognized predominantly in leukemias, lymphomas, and pediatric sarcomas, they are increasingly being recognized in all sorts of cancer types. A classic translocation that's described is the BCR Abel translocation in chronic myelogenous leukemia, or CML. So let's use CML as the prototype for an oncogenic translocation, in which the translocation occurs between two genes. And in the case of CML, this translocation leads to the generation of what's called a fusion protein. And CML is a type of leukemia. Specifically, it's a cancer of white blood cells. So there are increased white blood cells and white blood cell precursors in general. And it is linked to a characteristic chromosomal translocation between chromosomes 9 and 22. And we write the translocation like this. And now I'm going to pull up an example of what that looks like here. So as you can see, this segment of chromosome 9 and then this segment of chromosome 22 have essentially switched places. So now this part of chromosome 22 is down here on chromosome 9, and then you can see this part of chromosome 9 now part of 22. And so as a result of this translocation, the ABL gene, seen in blue here, becomes fused to the BCR gene, shown in pink here. And this creates a new hybrid gene, which we just call BCR-ABL. And this new chromosome here is called the Philadelphia Chromosome. And just sort of a fun side fact, it's actually called the Philadelphia Chromosome because it was first described by two scientists from Philadelphia, Peter Noel of University of Pennsylvania and David Hungerford of the Fox Chase Cancer Center. So this new BCR-ABL gene is constitutively active and produces a tyrosine kinase as its protein product. And it's this tyrosine kinase that in turn activates a cascade of proteins that control cell division and thus lead to increased cell growth. The BCR-ABL gene also produces a protein that inhibits DNA repair, furthering the development of genetic mutations and genomic instability. All right, so now that we've learned in a little bit more detail about what exactly an oncogenic translocation is, how do we then detect these types of oncogenic translocations? Well, these genetic mutations lend themselves to detection by either a karyotype, cytogenetics such as fluorescent in situ hybridization or FISH, or you can detect them by Rt-Pcr, which stands for reverse transcription polymerase chain reaction. So let's review really quickly a karyotype, and I'll bring up a picture of what that looks like here. All right, so here is what a typical karyotype looks like. It basically just shows the number and appearance of chromosomes in the nucleus of a eukaryotic cell. And so to get a karyotype like this, first a sample has to be collected from either blood, bone marrow, amniotic fluid, or maybe placental tissue. And then next, the dividing cells are cultured on a special sort of media and treated with a chemical that halts the cell phase in metaphase, which is when the chromosomes are most compact. And then next, the cells are treated with this hypotonic solution that helps to make the nuclei swell and allow better visualization of the chromosomes before they're treated with a fixative onto a glass slide. And then once fixed, the chromosomes are treated with a trypsin followed by this GMSA dye, which stains adenine and thymine. And then you get this characteristic banding pattern, which allows the cytogeneticist to tell one chromosome from another. And so karyotypes are useful for also diagnosing chromosomal imbalances in autosomal trisomies like trisomy 21, or say a sex chromosome disorder like Turner's, where one of the X chromosomes is actually missing. All right, so let's make a little bit more room here and we'll bring up a picture of what fluorescent Situ hybridization or FISH looks like. All right, so here is an example of FISH, or fluorescent in situ hybridization. And so FISH is just used to detect and localize the presence or absence of specific DNA sequences on chromosomes. And so as you can see from this picture here, you see little bright green spots and then little bright red spots. And these little spots represent fluorescent probes that bind to only parts of the chromosome with which they show a high degree of sequence complementarity, or what is called homology. And so if you look here, you see a little green spot right next to a little red spot indicating a possible translocation here. And this picture is taken with fluorescence microscopy once the probes are bound. And then finally, let's briefly review Rt-Pcr. So Rt-Pcr is just a variation on PCR, and it's used to detect RNA expression. And it detects the gene expression through the creation of what's called complementary DNA, or cDNA transcripts from RNA using the enzyme reverse transcriptase, which just reverse transcribes the RNA back into DNA. And the RNA transcripts from tumor cells are used as the RNA templates, and then the expression levels are analyzed with PCR.